Chapter. Share screen. And because we got a great show today, right? Complement. Know you've been waiting on this one, right? The good thing about the complement is all the numbers are numbers. Okay, so it's real easy to kind of keep up with, and it's only three different ways it works. So you have to get that, and it meshes all together at one point in the chapter. So we'll take each pathway separately, and then we'll combine them and show you how they're all going to work together, how they are ongoing working, and what's common about all three. Then we'll get into how you control it. So what are the controls that are in place to control complement? Then we'll get into the disorders of complement, like what could go wrong, what patient could have issues. And then we'll finish up with testing for complement activity. And we had a great lab for this, and it was really fun because it was like, here's some red blood cells, and we take somebody's serum and we put it in there, and complement just starts lysing these cells, and you measure that lysing. It's called CH50, so it's complement half-life 50, I think is what it stands for. They quit making it, so we couldn't get it. We couldn't find it to do. So you're just going to have to read about it, see about it, and we won't really have a lab about it. But it was really fun because we got to use the spec, and we were going to buy a new spec for just Immunology lab, and we were going to get this test, and then they discontinued the test. So evidently it wasn't a big demand for the test. You could look at it that way. And these cells only lasted, like red cells do, only like 30 days. So you couldn't like keep this kit like for months, and if you needed it, you could use it. So it was really, really, and I guess the easiest way to say it is complement disorders are rare. Okay. So we're going to talk about complement and the way it works for us, but when we talk about people that could be having problems with complement activity, it's very rare. So that being said, we can start on complement. So to overview, which I just gave you, we're going to look at the pathways. So there's different pathways. There's three. How do we control those pathways? The complement receptors. This should sound familiar. When we hit on this, you're going to put your memory caps on, because I'm going to ask you for some memory to come back on some of these receptors. Biological manifestations of complement activity. What does it actually do? The disease states when you have complement disorders and complement deficiencies, and can we detect those abnormalities? So we had a great test. I love doing it every year, but couldn't get it this year. So I have to say I'm sorry for that, because it was fun. It was fun lab to do. Complement is a series of more than 30 soluble and cell-bound proteins. So you have these circulating. So get the picture of what complement is. In your blood, these complement proteins are circulating. They're waiting to be activated. Okay, so right now they're circulating inactively in your body. As soon as you get a call for them to activate, they go through their process and everything just goes. So that's kind of the way it works. So they interact to enhance host defense against foreign cells. So they do chime in when it comes down to, as you've seen in all the chapters, you keep mentioning, they'll throw complement out there. Complement. This activates complement. That activates complement. So now we're finally going to get to complement to get this, finish our story before the test. So most plasma complement proteins are synthesized in the liver. So coming from our liver, there's one exception or two exceptions here. We got C1 components, which we're about to introduce in a minute. They come from the intestinal epithelial cells. And then factor D, which we'll see as we get farther into the third pathway, they come from fats. Okay, adipose is fat tissue. So a great multiple choice question, right? All plasma compounds are synthesized in the liver, except, boom, put one of these in there. C1 or factor D. White blood cells are an additional source of early complement components. C1, C2, C3, C4. And most of these proteins are zymogens that are converted to active enzymes. So zymogen would be an inactive enzyme. And we're going to start telling the story of how these go. So we got C1 is from where?
Intestinal epithelial cells.
Or white blood cells, right? And then we got factor D from adipose tissue. Okay. And then 2, 3, and 4 will be from white cells. Their main function is they act as opsonins, which means they help opsonization. They're the ones that put the salt on the antigen. Okay, so they coat it, and once it's coated, then the phagocytic cell is more likely to engulf it. So these are definitely opsonization. That's one of their roles. That's probably the main role. Okay, so when we talk about this is going to be one in particular. It's going to be C3b. I'll get to him in a little bit. Metabolism and clearance of immune complexes is enhanced. So basically, if we want to get rid of dead stuff, if we want to get rid of the waste, okay, complement is a great way to increase the chances of it getting removed. Okay. And there's a really neat one later in the process where we're actually going to see that erythrocytes play a role there. And I know you probably don't think about erythrocytes grabbing hold of an immune complex and circulating it through the body. The complement allows that to happen. So I know y'all are in the midst of hematology thinking. So we always think of what, erythrocytes, all they do is like transport oxygen with hemoglobin, right? That's their only role. Or they aid in the clotting off. Y'all ain't got the coag yet, but clotting off when we have a bleed. They also play a role with us in immunology because they're going to help circulate some of these complexes and get rid of them. Increased vascular permeability, that's going to be parts of complement. So when we first draw the complement pathway, we're going to say, oh, that one goes off here, and we really don't know what it does yet, okay? So we're going to split some of these proteins into two forms. We're going to call them A and B. And you're going to see that A, a couple of A's here that come off are going to increase permeability because they're involved with inflammation. Okay, recruit monocytes and neutrophils to the area. Okay, so that's good. So they're going to be those cleaving of the proteins, actually going to be attractants. They're going to increase vascular permeability. You are very familiar with these two terms, right? We've been talking about those since day one. They're also going to trigger secretion of immunoregulatory molecules that amplify the immune response. So as we said, complement plays a role in this whole symphony or orchestra of what's going on every day. There are 3 pathways: classical, lectin, and alternative. So these are the 3 you're going to focus on. These are the 3 we're going to bat around on the test on Wednesday. Is you're going to have to know the pathways of all 3 of these. We're going to go through that today. And that's the main part of today's lecture, is getting this in your brain. Like, get this down so you can play with these pathways. And they're fun. You can draw these out. You can draw them all 3 on one piece of paper. It's not like you're going to have 10 sheets of paper. You can draw them all out. It's pretty easy. But we have multiple pathways can be involved, so it's not like, oh, there's one way this is going to happen. They could all be going right now in your body. They all could be activated and running complement proteins in activation. And the end product is they're going to lyse the invading cell. So we have some that are going to, some parts of it are going to enhance phagocytosis, but they're all going to lead down to what we call lysing. So they're going to end up destroying the cell. Okay, so they're all going to have different ways they're activated. They're going to have all different kind of players, but they're all going to come into kind of a common way of destroying the cell at the end. Y'all with me so far? Yay? Yay? All right, so let's get busy on the classical pathway. And there it is. Simple as that. Looks easy, huh? So we have the classical. It's broke up into three kind of sub components. We have recognition, we have activation, and we have the MAC, known as the membrane attack complex. Okay, so this is going to hit the membranes and going to destroy the cell by lysing the membrane of the cell. All right. Y'all okay? Y'all distracted by noise?
Yeah.
I think that was against what's the rule? I was supposed to group up outside the classroom. Guess it's okay. All right, here we go. Back to the three stages: recognition, activation, and membrane attack complex. So in recognition, right, you see some players here. This is the only kind of drawback to the numbers, is that we start with C1. Okay, we start with C1. I'm just going to give you kind of like the flow of this to begin with. We got C1. This is the way you're going to be memorizing it the next couple of days, right? C1 goes QRS, right, together for recognition. And then they throw a C4 in there. Just, why not go to C2 here? I don't know. I didn't draw this up. I didn't make it up. But C4 comes before C2, and C4 comes before C3. If they'd have just done it 1, 2, 3, I guess it would have made too much sense. Okay? But we had C4 coming in, but you got to go C1 QRS, C4 and C2 come together. That activates C3. Okay, so this is kind of like known as C3 convertase. So we get C3 combines right here with C4B2A, and we'll do these numbers in just a minute. That activates C5. C5 is then plays a role in binding the C6, 7, 8, 9 to the membrane. That's the membrane attack complex, and all those together we form a hole in the cell membrane, okay, and destroy the bacteria. Okay, so it's really neat. That's just the intro. Is just put the numbers in the list first. C1, C4, C2, C3, C5, C6, 7, 8, 9. Okay, and that's it. No, that sounds like, oh, that's nothing to remember. That'll be easy. We got it. You've made a big deal out of this for nothing. But we're about to break it apart and show you all the pieces. So, how do we get this activated? This is the classical pathway. So the classical pathway is activated by either IgM, one, right? It's got those 10, arms. So it can be one IgM, or it has to be two IgGs. Okay, so that's kind of how we get it going. And those two IgGs have to be pretty close to each other. They can't be spread way apart and think that complement's going to recognize that they're there on the antigen itself. So these are the antigen immunoglobulins that have attached to the antigen. So their Fab region is what? Attached to the antigen. And that leaves their FC region to activate complement. All right? So you see the activation, and we break out the IgGs, and we say it's 1, 2, and 3 of the IgGs are all play a role. But IgM's by itself, so we'd have to have a couple of IgGs and a couple of IgGs. And then IgM is the most efficient due to its 10 arms. Okay? So that's kind of the start to the classical pathway. Classical can also be activated by some things we've already talked about. We've talked about, have we talked about C-reactive protein? I don't think, have we? As an acute phase reactant, did that come up yet? Yeah. Okay, good. All right. Certain viruses can activate it. Okay. Mycoplasma, gram-negative bacteria, that comes back up again. Okay, and some protozoa. So there's another list of also activated substance. So we got the immunoglobulin, right? We think about classical, we think about IgM and IgG, but it can also be activated by some of these other things, viruses and gram-negative bacteria, definitely C-reactive protein.
So those activate the pathway, or the pathway activates them?
Also activated by substances that bind complement directly.
And those bond different.
Yes. Okay, so these won't need the antigen present. Okay. These are hanging around. These are the antigens, basically. You want to think of the virus and the bacteria and the protozoa and the mycoplasma. Okay. So mainly those can activate. And that's what we're going to see about all three of these. There's different ways of getting these things started. So the classical got named the classical because it was the first one described. Okay, it's probably not the most used one of the three. The alternative is probably the most used of the three. But yeah, so don't get hung up that the classical was, you know, this is the way it happens all the time. But we're going to go through all three, but don't get where you're zoning in like the classical must be the best and brightest, but it's probably not as much as the alternative. And the reason being is the alternative is going to be just triggered by a bunch of other stuff. Doesn't have to be the antigen itself. All right, so here comes, this is a great picture from your book. And your book has this laid out. It's really nice. You know, I could never draw it this well. But it's the C1 that comes in, and this is C1, okay? It's the three subunits. The Q, R, and S are subunits, but gosh, they're a bunch. It's not just one of each. Okay, so we have one Q, okay? So this weird looking thing that kind of looks like an immunoglobulin coming in upside down, okay, is the Q. We have two C1Rs, okay, so those are, which ones? I think they're either red or the blue. The blues are the Rs. And then we have two little red molecules there are the S's. Okay, so this is a whole complex comes in. And look what it does. What it does, these are two IgGs sitting on the antigen. Okay, so what it does is comes in and binds to the Fc region of the immunoglobulin right there and right there. So it's really a complex itself. So it has to come together. Those C1 QRSs have to come together and form first. Okay, so you have those separate, they come together, they make one big complex, subunits, and then they bind to the immunoglobulin that is attached to the antigen. And this is that distance I was telling you about. These two IgGs have to be close enough that the one C1 can get to both. Okay, it can't activate. If it's just hanging on to one IgG, it's not activated. It has to be two. So you can see if that was an IgM sitting on the surface, we could get this. It would bind to the one IgM. It's got two Fc regions, right? So that's the activation, or the recognition. Sorry, the recognition has to occur first. Now what happens after that? Here's the C1q recognizes the Fc region of two adjacent antibodies, what we just said. At least two of the globular heads of the C1q must be bound to initiate it. The zymogens, which are inactive, right, C1R and C1S, are then converted into active enzymes, and they bind to the C1q. The autoactivation now of C1R results in the change that takes place in the C1q as the C1q bounds. So that's just words of what we just saw and tried to explain to you. When activated, the C1R cleaves the thioester bond of C1S, which activates it. So basically, you got the C1q comes in, binds to the antibodies. That allows the C1R and C1S to come in and bind. And once they're activated, they are doing some activity with each other. Okay. So now we're ready to move on. So hopefully you got that the recognition phase is that C1q comes in, binds the immunoglobulin Fc regions. You got to have two bound heads to it. I think it has 6 heads, but it has 2 of them are bound. And then C1R and C1S come in and bind to the C1Q. So that whole complex is the recognition unit. So when the C1S gets there and it's activated, the recognition stage ends. So that's just the end of our recognition. I'm not so worried that you're moving between each set, but I think it just helps keep it straight. Then we go from recognition to activation. And this is where all the fun begins. Okay, so the fun begins is we've got a C4. It's by itself, okay? You may want to call this inactivated C4 because it hasn't been activated yet. It's circulating through your body. And when it sees that there is a C1QRS sitting here, right, then it will cleave. This cleaves to C4. So the C4 molecule is cleaved into 2 portions, okay, called the C4A and the C4B. So anytime we cleave something, we're going to cleave it into an A and B. Okay? So the C4B, okay, as you see it, now cleaves who? Cleaves the C2. The C2 gets cleaved into C2B and C2A. And here's another weird thing because we do B and A here, but it's just part of this setup. You just have to know that the C4B and the C2A come together and get on the membrane of the actual molecule. See, so here's the activation, comes over, and then it lays down onto the membrane of the cell, or the bacteria, or whatever we're calling the antigen at this point. Okay? So far, so good. So just run this. You're going to have to, go through it the next couple of days. C4 cleaves into, and then we're going to talk about these. As you see, C4A is going off somewhere, and C2B is going off somewhere in the circulation. So they're done with this part. Okay, they're going to play a role outside in some other areas of the body, but not here. Okay, they're done. So that's laying on the membrane. That together, we change its name. We finally got it straight. We said it's C4B and C2A combined right next to each other. Then we can rename that as C3 convertase. Okay? So that lets you know it's going to convert. Now that we got C4B and C2A together, it's going to convert C3. And C3 is the big player. Okay, so you're getting introduced to the big player. The big player into the complement pathway is C3. Okay? C3 is in all pathways. Okay? So the classical pathway is done. Everything from here in is going to be similar to everybody else. Okay, so this is the unique part of the classical pathway. Okay, so we're not going to end. It's just telling you here that the activation unit ends when we get C5 convertase. And we're not there yet. We got to deal with C3 first. So here's how we deal with C3. You remember we got C3 convertase sitting there on the membrane. And here comes C3. C3 molecule comes in. Okay, it recognizes this convertase. It converts itself into C3b, which lands on the membrane too, next to the C3 convertase. And then C3a goes off. So C3a is going off, C4a is going off, and C2b is going off. So we got 3 stories to tell later as they leave off. Well, so you see through C3b set down real close to C4b, C2a, and now C3b together. That gets its name changed to C5 convertase. Because it's going to convert C5. Almost like a Harry Potter book, isn't it? We're just telling a nice story.
Yeah, but this is confusing.
So if C3 is in every pathway, do you have to go through this first step first?
No, we're just going to pick up those other pathways differently with C3. Okay. Well, C3 is just a player in all of them. Okay. But yes, we're going to get to the rest of the stories. All right, so C5 convertase, and once we get this one done, then the others kind of flow easier.
Is there a YouTube video on this?
There's YouTube videos, of course, yes. And I can probably show you one in lab today. It's kind of cute. It's an old one. It's just real quick, but it's a student-done video from another school. There's handwritten tutorials. I don't know if y'all dove into those. Like he does this too. He'll do all three pathways, handwritten for you. But it's one of those things. If you were drawing, it's like a Krebs citric acid cycle, but without all those weird names. You're just going to make a pathway on your paper, draw your arrows, and then put a blank out where everybody's going, and then just get where you can name them all. You just name through what we're doing. And then, of course, there's going to be multiple choice questions, so you're not going to have to draw it. You're not going to have to fill in the blanks for me.
Like the best way to study this, or how?
Sit down with about 10 little balls or whatever and play. If you got like 10 tennis balls. No, tennis balls. Sorry, y'all are smirking. Yes. Do something physically, like get something in front of you that do it. And then tell somebody the story, what's going on. All right, so we're getting there. So you see we have C5 convertase. Here's C5, kind of comes in like C3. It splits into C5b and C5a. So C5a is went and joined C4a, C2b, and C3a out somewhere to be told later. Eventually we'll talk about. So now we have C5b lands on the membrane. So we're stacking the membrane up with all these activated proteins of complement. So we're kind of building an airport, the way I look at it. We're building an airport for the MAC. The MAC is about to come in and start laying down a runway, and then we're just going to bomb the bacteria and put a hole in it, okay? So here comes the airport. So the classical pathway, this is the membrane attack complex. As I said, we left the activation when we got C5. Okay, once we got C5 activated, then we move into the MAC. So the C5 converted to C5a and C4b, right? Now they went away, right? The process is initiated when C5 convertase splits C5. That's not splitting it into C4. That's put a 5 right there. You know if I have a pen. Do I have a pen? That's a misprint. Oops, can't draw, I guess. Oh, I guess I should have pushed the button. See if I can get back to it. That doesn't make sense, does it? Well, mistakes are always for.
I think you have to press this little arrow on the side.
Press the arrow on the side and make my ink.
Yeah.
Okay, everybody got it? Those on Zoom, we drew through we scratched out the C4 there, shouldn't be there. See, we're done with C4. It's C5. C5 doesn't split into C5 and C4. We have a mistake on the PowerPoint. C5b, C5a. C5b attaches to the cell membrane to initiate the MAC. So the MAC is everybody, C5b, 6, 7, 8, and 9. You see 9 comes in as multiple parts, and that's what actually makes the hole in the membrane. Okay, and it lets the membrane, it weakens the membrane, and then as any membrane, once you punch a hole in the water either leaks out or leaks in, and the bacteria can't handle that, and it's gone. So that's the MAC. Luckily, the MAC comes in at 6, 7, and 8, and then 9 finishes it up. And that's the membrane attack complex. And that ends the story of the classical pathway. How'd we do? Y'all got it? Y'all studied all weekend. It's just review, right? You'd already jumped into chapter, chapter is this anyway? 7. You'd already finished 7 reviewing for my exam on Wednesday. Way ahead of the game. Sound familiar? Sound good?
Would you make a test question where it's like the formation of MAC includes, and then the choices would give random numbers and letters, or?
I can't remember. I mean, it's not too hard with the way it goes. It's like going to be a lot of who's in the convertases, like who's C5 convertase, who's C4, C3 convertase, who's part of the MAC, you know.
So we should like write the pathway, write the C, all of that, and then like kind of define what they are and what they do.
Yeah, make sure you put in the convertase names so you know what they are. All right. Promised you another way of activation. The other one is the lectin pathway. What it does is it recognizes carbohydrates in the bacterial cell wall. So we don't even need an antibody. Yeah, we don't need an IgG, we don't need an IgM, we don't need C-reactive protein. We just need the bacteria. The bacteria can activate complement, as we saw with that list of activating complement. You know, we had all those other things. But this one's specific for lectin binding, okay? So what you're gonna find is that we have defense mechanism and infancy, in between loss of maternal antibody and development of full antibody. So the lectin pathway actually aids in that space of mom's immune system going away and baby's immune system developing. So you would make sense. So if you get a bacteria pop up and baby's kind of vulnerable at this point, the lectin binding pathway would actually activate the baby's complement system and get after it. So there's 3 classes of molecules. There's lectin, there's phycolins, there's Cl, ligand 1. Okay, and after cleaving, what these do, which you will see a very similar look to this lectin binding. The lectin binding, I don't know if it's on purpose. I don't know if the lectin just happens to take the shape. This looks a lot like the C1QRS. Okay. Just take that for what it is. It looks a lot like that, the lectin binding, because what it's doing is lectin's coming together and binding to the sugar in the molecule, on the membrane, same way as what? Same way as Q1RS would bind to the immunoglobulin stuck on the membrane. Okay, so it kind of has the same similarity, look, and sound to it, but it's just doing it a different way. It's not needing the immunoglobulin there. It's just go ahead and binding to the membrane itself without that there. And I erase that, so it doesn't work. That's not gonna work. How about an eraser? That's not gonna work. That's not the eraser. Is there an eraser?
Yeah, it's that little box that you're pressing.
Right here?
The little blue and white, yeah.
That one?
Yeah, it should erase all, yeah. There you go.
Yay. All right.
Oh, it's over and down.
Yeah, it didn't work. All right. This was here because somebody was using this as a dry erase board and we didn't have a really good way of cleaning it off. It's kind of leaving stuff up here. So this was their answer. They brought us a little dry erase rag. For those on Zoom can't know what I'm playing with, it's the rag that's hanging on the wall. So back to the story of the lectin binding is that lectin binds to the membrane, right, like this, and then that cleaves C4, okay? Cleaves C4, which cleaves C2, and then we go down the classical pathway again. So lectin binding is really simple. It's just a different way of getting C4 activated. The lectin pathway, we call it mannose binding lectin, okay? So mannan binding lectin, it's a acute phase protein. It binds mannose or related sugars to initiate the pathway. So we have a couple of things. The MBL, you're going to see that throughout your little section here. That's mannan binding lectin or mannose binding lectin. That's fine. You want to call it that. And then MASPs, which are mannan binding lectin associated serine proteases. So MASP1, MASP2, MASP3 play enzymatic roles. So these are kind of the new players. Every time we talk about a different pathway, we're going to introduce new players. Okay? So here we are. You know, oh, that was it. Sorry. Mannose binding. It goes on. It just continues back to the classical pathway. I'm thinking, I thought there was a picture. Well, we're going to get to we've got one coming. I'll give you the preview. This is the preview. We'll finish off mannose here. So here is our classical pathway C1QRS and the 2 IgGs. Here's the mannose in the membrane. And look at the mannose binding lectin. Okay, here's our MASP1, MASP2, and MASP3, which are mannose associated serine proteases, right? That look a lot like who? RNS over here. So, sorry if I'm in RNS over here. Okay. So then that looks similar. I mean, I don't know if it's on, you know, but it would make sense that this now would cleave C4, C2, and then we end up with C3 convertase, and then we go down into the common way. Okay. So that is the two that are very closely related, which are classical and mannose binding.
So is the lectin pathway just another way to get to the classical pathway?
It's another way to get through, yes. So it's a different initiation part, or initiation, right? Isn't that the first step? Recognition, sorry. Recognition step is mannose instead of IgG or IgM. So the alternate pathway. Now the alternate takes all that recognition out. We don't need recognition with the alternative pathway because we're just going to jump to C3. So that's the jump. Take out the recognition phase. The activation phase is half done. Well, it's right where it picked up, right? Activation of C3 starts the activation phase. So the bad thing about this one is it's got some players too. Different players come in with alternative pathway, alternate. And this can keep going and going and going because we don't need what? We don't need the antigen. We don't need the antigen. Okay, so C3 just gets activated. It can be activated on its own by the bacteria, the fungal cell walls, the viruses, the tumor cells, or some parasite. So it's just, hey, we needed something bound to those, like, hey, we needed IgG and IgM first. That's for the classical. Hey, we needed mannose binding lectin to get up there to activate C3. That's in the mannose binding or lectin binding pathway. But we need something, or we know something, let's put it this way, we know something's going faster than that. We know more C3 is getting activated with infection than it would need. It started before the immunoglobulin got there, or it started before these other pathways that were discovered. And this is it. So what happens is it just, C3 decides to hydrolyze because it's been associated with bacteria, fungal cell wall, a virus, or a tumor cell. So the C3 is almost like, I can do this on my own. I don't need all that other stuff. I see a need. And it hydrolyzes itself, or gets hydrolyzed, water's everywhere. And it hydrolyzes once it's activated into C3b, okay, which we know C3b, we're familiar with that. And C3b, we're going to see maybe in the video, we're going to show downstairs, start lab off, it starts the opsonization process too. C3b can just pop on the cell surface and all of a sudden, phagocytic cells come in, finish it. We don't need to keep going to the MAC. We can finish it without the MAC with C3b. So they all kind of play a role, they all kind of play this, but we get C3b is sitting on the membrane now by itself. It didn't need C4 and C2b, okay, to become C5 convertase. But we can get there, and what you're gonna see is that we're gonna get there by some other ways of molecules are gonna bind, mainly factor B, okay? Factor B is the first player that's different for the alternative. But factor B looks a lot like color coordinated, and the book does a really good job of color coordinating this. If it's red and it's next to C3b, who could that be? You remember? Let's go to our common. If it's red and it's next to C3b, who could it be? So factor B is really kind of like C4. This is what I'm talking about. Here's B activated, and here's C4 activated. They kind of color coordinated themselves. So it kind of plays that role. And then we get this proparidin down here that comes in and kind of plays like the C2 role. Okay, so those are our new, and factor D is in the mix. So we got factor B and factor D playing the role here. Factor D cleaves factor B into big B, little b. Yeah, we don't have a number now. We're just going to call them big B, little b. And big B, little a. Big B, little a goes off. We'll see what that does later. But that's the cleaving that happens when factor B comes in and recognizes C3b. So it's almost the reverse of what happened with who? C4, B, and C2a. They came in first to convert C3, right? C3 gets converted first, and then they come in separate. They're known as factor B and proparidin, okay? So here we are down here, all right? So we talk about this as C3b, big B, little b, and then that's an enzyme that that becomes C3 convertase. More C3 is cleaved, forming more C3b, big B, little b. And then we have a stabilization with proparidin here, okay? And that will continue to cleave C3b into C3 into C3b, C3a, until we actually get it to stabilize with another C3b attached to the proparidin, attached to the big B, little b. So we call this C3b, big B, little b, P, right? And that's going to give us the C3b, big B, little b, P, 3b. And we'll leave it right there. And that's C5 convertase. And then we continue on to where? The classical pathway and the MAC. Okay, so this is a key here to the difference in the alternative versus the others, okay? It's how C3b comes in by itself in this alternative pathway and starts it all off. So the players are factor B, factor D, and proparidin. That's it. Those are our new players that we've got. We'll need to know their role. And then how we end up with the C5b onto the membrane to finish the MAC is the finishing of alternative pathway. So there it is all together. Okay, so if you want to study and you want to look at it, follow this. Once you get the three done by themselves, you can come in and pick up and see how they all roll into the MAC if needed. Like I said, C3b can work by itself. Doesn't need to go all the way to the MAC. All right. Now, not to confuse you, we'll put the inhibitories in. How do we control this? Okay, so you got to know the way the pathway first, then these will make sense. If you don't know the pathway, these won't make sense. Okay, they won't. You've got to learn the pathways and then come to what inhibits the pathways. Because you won't know if you got C1 inhibitory protein, right? It disassociates C1R from C1S in the classical, and then from the C1Q. It also says the involved pathway is the lectin pathway. Do we have something that looks like the C1 in the lectin? Yes. Would this be inhibitory to that? Yes. All right. We have factor I. Factor I inhibits or cleaves C3b and C4b to keep them from continuing on. Okay, so it puts a stop to C3b and C4b, and those are both in the classical and lectin pathways, factor I. And really, let's focus factor H, cofactor with factor I to inactivate C3b to prevent the binding of big B. So anytime you see big B, okay, it's going to be the alternative by itself. Anytime you see who? You see 4B, that's lectin and classical, right? We didn't have C4 and who? The alternative. So that's a good way to just got to remember where they are, what they're doing. All right. And then we have an S protein, the vitronectin down here that prevents the attachment of the MAC. That would be all of them. So anytime you're dealing with the MAC complex, it's all pathways. Anytime you're dealing with C4, it's the first two. Okay. Anytime you're dealing with factor B or factor D or procaritin, that would be the alternative. You got a lot so far, right? So this is where it shows where these all come in. It shows how they display. So this is just kind of helping you worry with factor H. Remember, factor H is the alternative that prevents binding of big B to C3b. That would stop it right there, wouldn't it? If we can't get factor B onto C3b, that ends the whole deal. So factor H comes in and blocks big B from binding to C3b. So it stops the alternative pathway. This is in a little section of the book. It's on page 99. Okay, right underneath the table that talks about the inhibitory factors. And these are known as DAF, which stands for decay accelerating factor. The MIRL, where is Mural? And MCB, where are those? I was going with C4 big B P and C1R membrane cofactors. Just want to tell you where they were.
The membrane cofactor protein is on 99, right above DAF,
Yeah.
but I don't know. It's on page 102.
102, thank you. There we are. There they are. Okay, so you got receptors on cell membranes and complement. What do I want you to know of this? I think we're okay. I'm not. What these are is how does the so basically what we're getting is these are the receptors on the membranes, and what, oh, I know what I'm going to tell you. Notice these have what? Our cluster differentiation numbers, right? Remember those? Okay, so these are on the actual membranes of the cells that we're looking at. Red blood cells, neutrophils, platelets, monocytes, endothelial cells. Okay, so they play a role in binding C3b and C4b. Okay, these complement 8, C8, C3b, C4b. Okay, so these are controls with the cells themselves that would actually interfere. The function, they disassociate C3 or C2b or big B, little b from the binding site. Okay, so there's some control here, is what I'm getting at. Prevents insertion of C9 into the cell membrane. Why? Because it's working on the C8. It's binding. These receptors are binding away some of the complement components to control the finishing of the complement. The complement doesn't finish if it doesn't get there, okay? So that's kind of a big picture, but that's what I wanted you to remember, is see that these are CD 55s, 59, 46. Not so much as they're those numbers, but they are CD receptors on the cells here that we're seeing, and they bind parts of the complement to get them out of the way. So if you want to control the pathways that we just talked about, you take these away, take these components away. Take C3b away. Yeah, that stops stuff. Take C8 away. Take C3b away. Take C4b away. And we see that. And you see more of these.
So are these like disorders or just regulation?
No, these are roles of the other cells in the complement pathway. So these cells, red blood cells, neutrophils, where we started talking about the very first cells of the, here are the red blood cells. Monocytes, macrophages, they're in there doing what? Binding some of these components to slow down complement, okay? All right, so this is to finish the story. We're going to wrap up. We've got a couple more things to say, and I'll be done. One being that what is the whole idea of the complement? The inflammatory response. All those ones, a couple of those that we said went on, right? There's a couple of them. The CAs, right? C4A and C3A go off. C5A goes off. They amplify the inflammatory response. C3B, recognition of the phagocytic cells, so they can actually get on the antigen by themselves, not have to go to the MAC. We got B cell activation. So this whole complement activation plays all the things that we've talked about already. Anaphylaxis toxins are there. Opsonins are there, and chemotaxins. So these are how they affect. So all this sounds familiar, should. And then we can have harm from complement. Complement can be activated on a large scale, the gram-negative septicemia. Tissue necrosis from a heart attack. You have a heart attack and the heart got like no oxygen, so the tissue starts to break down. That can activate complement during a heart attack, get tissue necrosis. And then lysing of the red blood cells can occur. And then that's our test that we talked about at the beginning with complement measurement. Lysing of the red blood cell is when it's activated. So if the antibodies get on the red blood cell, complement can do that. So when we talk about blood bank coming, when we talk about transfusion reactions, when we talk about lysing of the red cells, this is how it's happening, okay? So when your patient all of a sudden is having bloody urine after they've had a transfusion reaction, it's because they lysed all the red cells, they're in the kidney, and they end up in the bladder, and they're being expelled. All right, so we're gonna save this. This is kind of how we connect all the different deficiencies, and we said these were very rare, but we connect these to kind of like, if we didn't have one of these, what would happen? We said C3 was the main player. Y'all should see that now. Severe recurrent infections and kidney infections. That's our main one, okay? All the others are minor, but you can see kind of similarities between them. Like Hey, we get pneumococcal diseases, Neisseria, those that are in micro, sorry. For those that aren't. Okay. All right, and then we finished up with detecting it with our story of complement CH50 on how we detect it. Okay, we don't have the lab to do that, so we'll leave it there. Think you got enough? Okay. We'll see you at 12:30 for case studies to help maybe go over this a little bit more on chapter 7's case study. But that's it. All right, Zoom, we'll see you at lab.